Okay, and so this is primarily based off of my experience going through many MRIQC reports in order to do quality control for my lab's data sets. And so it's just based on my experiences doing that. So what you will learn in this talk is hopefully you will be convinced to do some form of QC on your data. You'll be convinced of the importance of QC. You will see what it looks like to do quality control using MRIQC, and you'll have some resources for evaluating scan quality. But I will not be teaching anything about MR physics, nor am I able to answer any questions about that. And one thing that's going to be a consistent theme across this presentation is that there is no definitive exclusion criteria. With rare exceptions, there are no definitive criteria for exclusion. So what might be perfectly fine for one type of study, for one type of scan, might not be fine for another. And so your exclusion criteria will always be dependent on the data that you're collecting and the types of analyses that you want to do for that. So before we get into actually doing the quality control, I just want to motivate why we should do QC in some sort of standardized way, because it's not the most fun thing to do. It's a little bit boring and can be time consuming, and I would love to skip over it. But they say garbage in, garbage out. And so that means that the quality of your analyses depends on the quality of your data. And this is, we know that, my animations are not working. That's okay. We know that motion is correlated with clinical variables and other variables that we care about, like age, clinical status, and sometimes even your task design. So your participants might be moving in response to a part of your task. We know that motion exacerbates other MR artifacts like ghosting. So it makes these other artifacts that we can't really avoid with the MR physics, it makes them worse. And even if we try to do some sort of aggressive denoising, even if we are able to remove the effects of motion and other artifacts, we also might end up removing true signal. So we are lowering our power doing that. And so this right here is a plot just showing from a relatively old paper showing the correlation basically between how much the participants moved and their age. So 8-year-olds, this is their average displacement in millimeters. So how many millimeters their head moved. 8-year-olds, that was about 0.4, and then that drops off to almost nothing by age 22 for a good participant. So if you're interested in things like development, this is really important, because any effects that you find about how the brain changes with age may actually just be attributed to motion if you don't do proper quality control. And you should be doing QC in some sort of standardized way, because the variability in the ways that different labs do QC can impact the reproducibility of our analytic outcomes. So a couple of years ago, Paul Taylor at the NIH did something similar to the NARPS project that Russ talked about earlier in the week, where they gave a data set to a whole bunch of different groups and said to the groups, do whatever you would normally do QC-wise to this data and report back to us which participants you end up excluding. And so what we see here is that there's a lot of variability. So each one of these labels down here is an analysis team. Each row here is a different subject, and so there's different subjects all across. And we can see that like some of the teams excluded almost every participant, and some of the teams excluded almost none. And a lot of that is explained by just the different types of analyses that different labs use, or different types of analyses that the labs were intending on doing, and different ways of doing the QC, more robust ways of doing the QC, and also just how stringent you wanted your QC parameters to be. And so there is any one of these could be right or wrong, depending on the goals of your study. And this is just to show that it's very highly variable. And so you can imagine if if I try to reproduce one of the studies that these teams did, but I don't know how they did QC, I might end up including a lot more participants or a lot fewer participants. And so I'm going to take a step back and talk a little bit about MRIQC for a second. So MRIQC falls within the NeuroPrep's ecosystem, the neuroimaging preprocessing tools ecosystem. You might have heard a little bit about this previous in the week, but this includes apps like software tools like fMRIPrep, dMRIPrep, smRIPrep. And essentially the broad goal of these tools is to create sashimi grade data, which I think is just a really cute analogy. So data that's minimally preprocessed, but safe to consume directly. So we're doing as few things to this data as possible, but we can still take this data straight from something like fMRIPrep and do some analyses on it. I will point out, though, that MRIQC does not actually do any pre-processing to your data. All it does is it takes the data in, it makes you some nice visualization and reports some image quality metrics, and otherwise doesn't do any processing to your data. That if you want to do data processing, that's something that fMRIPrep and these other ones here will take care of for you. So this is a bit of a roadmap for what we'll be talking about for the rest of what I'll be talking about for the rest of this talk. So we start all the way over here at establishing standard operating procedures for data collection and quality control. And then we go to how do we store and use these QC results. So standard operating procedures are important because it makes your data collection and your quality control procedures more reproducible, and also in that other people can look at your standard operating procedures and follow those, and more transparent, they can look at them and follow them. And it also makes it easier to do things like train new research coordinators, because instead of them having to watch you while you explain everything and they're trying to remember everything, and it's very overwhelming as a new research coordinator when you're trying to learn how to run the MRI scanner. If there's a standard operating procedure, then your new RAs can just refer to that easily. And then it's easier to write up your methods, because you can, when you're writing a paper, say, oh, this is exactly what I did. They can also reduce within and between researcher variability. So you know that everyone in your lab is preparing the participant in the same way for the MRI scanner. You know that everyone is looking through their QC documents in the same way. And so that can just make your within lab variability a little bit less. Documentation is good. It's helpful to remember what you were doing previously. And so when you're writing a QC standard operating procedure, you should cover data collection. So how do you prepare the participant to collect the best data that you can? How do you, if you are at a scan center where you are the MRI tech, how do you set up the scanner so that you can get the best data that you can? They should cover quality assessment. So how do you go through how in this case, how do you go through the MRIQC HTMLs and the visualizations that are generated by MRIQC? How do we go through those and assess the data? And in here, they should also cover what would make a scan ineligible, what would make a scan bad enough that you wouldn't want to use it. And then how do you store these QC documents? So do you do you add them to the participants.tsv file in your bullets folder? Do you make a different file? Things like that, so that, you know, in 2 years, when you have a new crop of research coordinators or the graduate student leaves, how do you find the QC that's already been done so you don't have to do it again, which is something I've had to do, and it's not very fun. So here are some examples of standard operating procedures. This is for a project called the Human Connectome Phantom Study that's being run by Oscar Esteban in Switzerland. And so there is a very extensive web page here talking about why they're doing this project, but also how do we do the recruitment? Who are we recruiting? How do we prepare the session room for participants? And then different things about the data management. So anyone who has any questions about this project can go to this website and note basically anything that they would need to know. And another example here is a much more low-tech version of a standard operating procedure that I wrote for running the scanner when I was a research coordinator. So just things that I needed to take note of and to double check, and then how do I run the scanner? Which buttons do I press in which order? What scripts do I have for talking to my participants and things like that? So your standard operating procedures can, you know, cover a range of level of description. So, like I said, your standard operating procedure should define exclusion criteria, which will vary depending on your population, what type of analyses you want to do, how robust your pre-processing tools are. They might include things like movement cutoffs. So are you going to exclude any participants that have an average movement of above 0.4 or 0.6? What kind of artifacts might you expect from your specific scanner? And what sort of artifacts can be fixed with pre-processing? Pre-processing can do a lot, but it can't do everything. And so it's good to kind of have an idea of the types of artifacts that can be fixed and the severity of artifacts that can be fixed by your pre-processing tools. And then you should talk about how you're going to resolve inter-rater disagreements. If you have two people writing the same scan and I think it's excellent and Kelly thinks it's trash, who do we trust? Maybe Kelly. Or do we have a third person then come in, or do we ask our PI and they're the ultimate decision maker? That is something that can be noted in your standard operating procedure as well. And also lastly, just incidental findings. So if you discover someone has a tumor, what do you do about that? Don't contact the participant, contact your MRI center. But having that written out is very helpful. And so I've put together some training resources here that I relied on when I was trying to learn more about quality control. Ben Inglis at Berkeley has a really great blog. It's a little bit technical. It's a little bit heavy on the MR physics, but he does have fairly good explanations of some of the common artifacts that you might find. My paper is a good resource, and it comes along with a supplement that shows some examples of the artifacts that I'll be talking about. My co-author on the Nature Protocol paper, Celine Province, has an excellent paper looking at MRIQC and fMRI prep. So it's 2 for the price of 1. And then NeuroStars can also be a good resource. And you can always check with your local MR center and see if they have any resources. Okay. So the first, moving on to our second step, our first step, but our second step, it's best to just collect good data from the outset, and then you don't have to worry as much. So pilot your scan sequences, make sure those are solid before you go forward and collect 100 participants worth of data. Use your mock scanner when needed for participants, for populations like younger kids or clinical populations. Ask your participants to stay still, remind them to stay still. That's pretty important. Most of them are happy to try to comply as best as they can. Make them comfy, spend some time setting up the scanner, check in with them to make sure that their legs are in a comfortable position. Monitor their movements while scanning. You can do this with different technologies that your MR center might have installed, or you can just watch their feet, and if their feet are moving, they're moving. And then check your scan quality as soon as possible after scanning. This can be important so that you can, if there's something wrong with the scan and you need that participant, you can ask them to come back and do that scan again. Or if there's something wrong and it's something related to your scanner, like there's some dust in the scanner, you can let your MR center know ASAP before it makes other people's data bad. And one thing that used to be kind of a thing that people were trying to do was these custom printed styrofoam head cases that basically filled in the gap in between the head coil and your face. So you would basically do a 3D scan of your head, send it off to some people at Berkeley, and they would then send you this head case that would pretty firmly keep you from moving, but later analyses showed that that didn't work very well. So now these are just relics, but it was a fun idea. Okay, Ariel talked earlier about bids. So MRIQC only works on data that is organized in bids. Organizing data in bids can be hard. It can be easy, but it's always ultimately worth it because there are so many tools that operate directly on data that is bidzified. And so, you know, Ariel showed this before of this kind of really ugly DICOM directory with, I wouldn't know what these scans are, what these files are because they're named with numbers and I don't know what those numbers mean. But then you can organize it into a really nice data set where I can say, oh, this is the data from subject 1, and they have an anatomical scan and they have a functional scan. And their functional scan was done during rest. And then they have these diffusion scans as well. So I think Ariel said that they'll be doing a different session on bids creation, so I won't talk about that too much, but there are a lot of different tools that you can use to get your data into bids. The next step would be to install MRIQC. Today, I've just prepared some example participants because the Jupyter Hub does not play very well with Docker containers, which is the best way to install and use MRIQC. You can just install on the command line with pip install MRIQC, but it does have some dependencies that are a little bit that you also have to install. So the best way to do it is either using a Docker container on your laptop or an Aptainer on your high performance compute cluster. I'm not going to talk about that right now, but if any of you are curious about that or want help setting it up on your computer or on your institution's high performance computer, let me know and I'm happy to help out with that. These containers essentially make it so that all of the dependencies that MRIQC has, they're just installed on this container on your laptop. And it minimizes difficulties with different OSs and different dependency versions, and it makes your life as the end user a lot easier. If anyone is familiar with Flywheel, it's one of the ways that data can be stored from the scanner. It's a cloud platform where the scans go up there after they're acquired, and MRIQC is installed as a gear on Flywheel. And then there's things like brainlife.io and neurodesk.org if your institution, if you have a large data set and your institution doesn't have a high performance compute cluster, or you don't have access to it, you can look into Brainlife and Neurodesk. And yeah, I'm happy, so happy to help you configure any of these later in the week. But for now, I decided that I wanted to focus on MRIQC and not on trying to get it installed on everyone's different computer with your different OSs. Okay, and so then the next step is to execute MRIQC. If you have used a bids app before, this will be somewhat familiar to you. So the command line is very straightforward. It's MRIQC, your bids directory, your output folder, and participant. Your bids root has to be a bids data set. Your output folder will usually be bids root / derivatives. You leave this participant as is, and usually this will take about 2 to 5 minutes per scan that you have. So for example, PNC has 3 functionals and 1 T1, and it was taking 7 to 8 minutes consistently for me to run MRIQC on my laptop. And so just an example using the file paths from the Jupyter Hub, we have MRIQC, my path to PNC bids, my path to derivatives, participant, and then my participant label is this first participant. And you just run that from the command line. So after you run that, each individual scan gets an HTML report that has visualizations that tell you something about the quality of the data. So we can see here all of our HTMLs. And then we also get a TSV, a tab separated file with image quality metrics like the frame-wise displacement. So these are things that MRQC, like objective measurements that MRQC calculates and provides to you. And then once you've executed all of the participant level once you've ran all of the participants through MRIQC, you can run the group level MRIQC, which essentially collects all of those IQMs from the different TSVs and gives them back to you in a nice visualization that shows the distribution of those IQMs across participants. So you can see like, oh, do I have any outliers? Did one person move way more than everyone else, or is everyone fairly tightly clustered? And so that again is a straightforward command line, MRIQC, bids directory, output directory, and then tell it that you want the group level. And then next, we're going to look at rating the reports. So part of the reports is this cute little ratings widget, which allows you to keep track, to rate your images in the MRIQC HTML itself, take notes about it, and then save these ratings to a JSON file. So JSON is just one specific type of file. It's a specific type of text file. So those get saved to your computer, and then you can later go through and look through those ratings, or pull them into a Python script and do more plotting, compare them with your co-rater and things like that. So let us start with looking at some of these ratings, and we're going to actually start not with the PNC data set, but we're going to start with another data set from Open Neuro that I ran, just because this is a better example. It usually doesn't take this. Okay, there we go. So like I said, each scan gets a report. There's one T1 in this data set. So there's one T1 report. This is an HTML, so it just opens in your web browser. For me, that's Chrome. I believe MRIQC works with most of your normal browsers. Just opens in the browser, and then you can scroll through it like so. We can pull up our rating widget up here. So we can look and we can, I mean, I haven't looked at it yet, so I can't quite determine the quality, but if I drag this here, I can say it looks excellent. And this is on a continuous scale from, I believe, 0 to 4. So doing something like this would be like a 3.7. I can record specific artifacts if I see any specific artifacts, or if I see anything that's not listed here, I can make a comment here. I can write how confident I am about this, and then I can download it, which then opens it. Yes.
One question. Is it possible to change the contrast? Because for visual rating, it might be more helpful to have it in a scale of gray, maybe?
It is not possible, but if that's something that you're interested in, you can go on to the MRIQC GitHub and post a request for that. There are different visualizations here. And so here is what we call a standard deviation image. No, this is a background image. So this is an image that's basically enhancing the background, so the non-brain part of the image. So we can look and see if there's any sort of background noise. And in this, in that sense, this is a bad example because there is no background noise. What we're seeing is a nice clean background with no noise. And then what we have here is just your normal view of the brain in grayscale. Yeah. A little bit zoomed in, right? So your scan will probably have a little bit of space on either side of the head. So this is zoomed in so you can really look more closely at like kind of this inner part. Sorry, I'm scrolling. I scroll through these kind of quickly because I know what I'm looking for. And so we have these plotted both on a coronal view and a sagittal. And if you run MRIQC with a specific flag -- verbose reports, you get this extended visual report that can be helpful if, say, MRIQC is crashing, or if MRIQC isn't executing the way that you think it should. These visual reports are more for debugging MRIQC rather than any sort of actual QC of the images themselves. So that would be your average T1 HTML is something like this. And now I will show you. will paste for me. We'll just open it up this way. So this is from the PNC data set. And so we can already see a little bit of a difference. There is, yeah, I guess that's showing up okay. There's a little bit of noise in the background here. So kind of like near the brain here, there's some like really light green speckles. That's not, I point that out only because it's a difference from the previous scan. That wouldn't necessarily be a meaningful difference. This isn't going to likely impact the quality of the scan. I think we can see the participant's ear right here. And again, this looks like a pretty good scan. And then we get to move on to to this mosaic view. And what I want you to note here is just the difference in the contrast between the white and the gray matter. So if we go back here to our other scan, we can see that there's a lot of contrast between our white and our gray matter. For the PNC data set, not so much. So that might be something that you're concerned about. This would be a lower quality scan. It's, in my experience, this would be fine for an analysis where you're only using the T1 for the alignment of the fMRI scans, but you would have, if you wanted to do something like looking at the cortical thickness, I don't know, you would probably have to look at that a little bit more closely. This doesn't look excellent. But I'm not, I am more of a functional connectivity person and less of a surface anything about structural. So yeah, you would take that with a grain of salt.
One quick question. Was that data rephrased? Did you give those images a fake face or did you remove the face?
Yeah, so these are refaced. Honestly, I don't know what that means. That's something that the PNC, the folks who curated the PNC data did. I wouldn't know the purpose of refacing your data after you've defaced it, but I don't know, does anyone else know? Yeah, I don't know. It might be somewhere in the PNC publication. But I will talk about defacing your data in a little bit, once I find where my slides went. The PNC data? Yeah, this is the RBC data that you've been working with. Right, it's been refaced. So I think they took the face off and then put something back. Usually, yeah, so that is an interesting thing to talk about. Is when you deface your data, you're actually losing some information, right? You're removing, so defacing, you have your MRI scan. MRI scans usually have faces because the scanner picks up that part of your anatomy, and that can be used to re-identify you. And so if you have openly available data and you have the face on it, then there's the threat that a nefarious party will come along, re-identify your scans, and your brain is protected. That makes it protected health information. And so say an insurance company could use that to raise your premiums or something. Essentially, it's a way to re-identify the data, which is something that we try to avoid. So typically you deface the data, which in a lot of cases just means zeroing out the voxels of where the face is. And again, I don't know why you would then reface the data, but I'm assuming it's not refaced with like their actual face, but refaced with like maybe an average of some sorts.
Question. Sorry if I missed this, but the data you were showing us, this is prior to pre-processing?
This is prior to pre-processing, yeah. Ideally, well, yes, you do MRIQC prior to any sort of pre-processing.
That's interesting because like using fMRI prep, usually we QC like in FSL, for example, or like we usually QC after pre-processing. So I'm wondering like why did we do it before?
Yeah, that is a really, really good question. And right, you might say, why would I want to do this multiple times? Right, because if you're doing it on the raw data, you probably also want to do it after fMRI prep has been run on it, just to make sure that fMRI prep did the things that fMRI prep is supposed to do. And the answer to that is that doing it on the raw data means that one, you catch artifacts that might not be obvious in the pre-processed data, in the visualizations that fMRI prep does of the pre-processed data, but might be, say, like something that's up with your scanner that you might want to look into before those artifacts get worse. Or they could be like more subtle things that just would be masked by the fMRI prep processing that you do want to note about in your data. Does that answer your question?
Yeah, it does. I guess, yeah. I was wondering whether you then do this 2 times, like once raw and then once like during fMRI prep.
Yeah, so the nice thing about fMRI prep is that it also gives you these nice visualizations. So they'll give you very similar visualizations just after it's been processed.
Just a follow-up from that because you were mentioning fMRI prep. I'm guessing that for fMRI images, the motion values are going to be similar for fMRI prep and MRIQC. They might not be exactly the same values. So do you know if they can like differ a lot, like the MRIQC motion values and the fMRI prep motion values?
That's a good question. My guess would be that they use the same function to calculate the, like, say, the frame wise displacement. A lot of the code from MRIQC is like a knee preps workflow, and so like I think it borrows a lot. I know at least the visualizations, they're all the visualizations for fMRI prep and MRIQC are all generated by the same basic code, and so I would assume that they're very similar, if not the same.
Okay, thanks.
I'm sorry, I'm just having some technical difficulties finding my notes again. Does anyone else have any questions while I try and figure this out?
So I guess thinking broadly, in the context of like an entire, say like resting state pipeline, if you're taking QC notes on like the raw data and you do some pre-processing, and then you do some more processing, do you have like a recommended workflow for keeping track of all of your different QC notes at different stages? Like, do you recommend like appending the original like JSON so that way it's all in one place? I know that you've had a lot of experience doing this. So.
Yeah, that's a great question. I would include them, and I believe this aligns with the bids specification. I would put them all, so you know the participants.tsv
Mhm.
file that's in the base of the bids directory, I would put it in there.
Yeah.
So you can do like MRIQC ratings, and then you could do fMRI prep ratings, and then you could do like even more post-processing ratings. Something that we talk about a lot in the paper is you should be doing QC at every major processing step. It's kind of arduous, which is why we advocate for using MRIQC, because it is a little bit more streamlined than going through every, say every time point of the fMRI image. Does anyone have more know how to use Google Slides any better than I do? Yeah, and that is where I have all of my examples. Right, and so I can get presenter view back, but here. So the slides are here, and then if you go here and you can open up.
But you have presenter view already. You can switch the really quickly. You can swap. Do you want the presentation here?
Presenter view, the presentation notes here. Yeah,
she has.
I've been doing that, but it's been opening it with the presentation notes right next to the.
If I just got my laptop, I'm gonna get the notes on it.
Oh, I'm good without the presenter notes. The presenter notes just have the links to all of my example files.
That should-
Yeah, okay, cool, thank you. Oh, thank you, that works. Excellent. Okay, so that was your T1 images. Your T2 images will look very similar. PNC does not have any T2 images, but we can look at some other data set. So as part of my paper, we also distribute example HTML reports for the IOMIC PIOP data set that's on Open Neuro. It's fairly high quality. We just share this so that other people can look at the images as well. Oh, it doesn't have T1s. It doesn't have T2s either. T2s look a lot like the T1s. If you have any examples that you want to look at, come find me later. You're looking for basically the same things. And then your functional images are going to look a little bit different. So in this visualization, what we're looking at is the standard deviation of the variability of each voxel. So the standard deviation of each voxel. And so we can see that the eyes are very variable because the eyes are always moving. Like even if you're doing just like a simple task, your eyes are going to be moving. And so there's going to be a lot of variability up here. There's also a little bit of variability here around the edges of the skull that can be related to motion. And what you will also a lot of times see like this artery here. You can't get rid of that physiological motion. Your best participant still has to breathe. So that's unavoidable. Same with eye movements. Otherwise, this looks like a scan that you would want to have. You can see that the ventricles tend to be more variable than the rest of the brain. And what you want to look for is nothing inside the brain that shouldn't be inside the brain. You can see, again, in this visualization, we're looking at the background of the bold time series. So that, again, this is just upping the contrast in the background. And we can see that there's these spikes coming out from the brain. This is related to a specific type of MR artifact, but these don't appear to be super severe, and you can't see them. You can't see any distortions in the bold image. So you're probably fine. You might notice that right now I'm going back and forth between these two images. So you might think like, oh, I'm just going to like scroll through these in order, when really it's kind of a scroll back and forth process. Like I might think, oh, this looks fine, but then I scroll down here and I say like, oh, this looks weird. Like the fact that this top of the brain is lighter than the rest of the brain. You can see how, right, like see how much brighter like here is compared to the rest of the brain. I might find that suspicious. So I might scroll up here and see if anything looks off in this background image, and see if anything looks off in the standard deviation image. In this case, this is just another normal MR physics artifact. It's called intensity non-uniformity, and it's basically because of this part of the brain being closer to the coil. And this is fixed really easily with fMRI prep. So not something to be super concerned about. If you have a multi-echo data set, you'll get one set of these visualizations for every echo. So then that's nice to see, like, do your later echoes look better or worse than your earlier echoes? Another thing that we get in the fMRI MRIQC reports is these carpet plots. So up top, we have a plot of how the percent of outlier voxels, DVARs, and FD. I primarily look at this last one at FD. This is frame-wise displacement. And what you're looking for here is any large spikes in movement. And so we can see this participant moved, the most they moved was 1.22 millimeters, but their mean was very low at under a millimeter. Like, this is a good participant. If you have a couple of large spikes, that is sometimes okay, because that can be dealt with really easily by scrubbing if that's something that's amenable to your analysis. Again, it's hard to diagnose any issues just based off of this plot, but if I see that, oh, if a participant has really high movement, I might be more suspicious of these images and look again more closely at these images. And then this is what we call the carpet plot. It's from what's his first name? A Powers paper in 2012, where essentially all of the voxels in the cerebellum are taken and plotted on this y-axis across time on the x-axis. And so you can see how each individual voxel, each individual voxel is one row, changes across time. So we can see that the carpet plot, fluctuates based off of these movement spikes right here. Hard to diagnose anything definitively from the power plot, but if there's anything that looks funky about this, I'll go up here and look. And then this is a very similar visualization to this mosaic view, except it's zoomed out, so you can see more of the space surrounding the brain. You can see the skull a little bit better, and you can look to see if there's anything happening outside of the brain that shouldn't be happening. Any questions? Okay.
Was that only one run that you showed?
This is one, this is one,
run?
one task. Yeah. So then for this participant, I can also go and I can find, I can grab their next task.
How do you interpret those blocks of white effect?
Sorry?
How would you interpret the block of very high intensity?
Right here, because it follows this this movement spike right here, I would say that the participant moved their head. Cool. So that was a pretty good looking scan. And then I picked this one out of PNC of the data set that you've all been working with, because this participant has incredibly high motion. And immediately, if you've looked at some of these before, you know something looks weird because this is just very fuzzy and blurry. There's a lot of yellow around the sides of the head, which means that those voxels are changing a lot, which is indicative of motion. It's just really blurry. You can see here also very blurry. Even the spikes off of the head are blurry. And then when we scroll down here to our frame-wise displacement, we can see that their maximum was 28 millimeters, which is super high. Their mean was only 3 millimeters, but that's 3 millimeters is still pretty high when you're looking at like one rule of thumb is like 0.2 might be acceptable for a like normal adult population. 0.6 might be fine if you're working with kids. So something like 3 millimeters is extremely high. I would probably exclude this participant. Any questions? Cool. And then, yep.
So have you like encountered a participant having like poor QC in this context, like for the unprocessed data, and then their data undergoes processing, and then it like looks pretty good, like similar to other people's? And then if so, how do you go about deciding whether to exclude them or not, at least like from your experience?
Yeah, so I have experienced that before, and we've gone with if it looks good after pre-processing. This, I can fairly confidently say that this participant probably wouldn't look very good after pre-processing, and just based off of what I know with motion and the types of analyses that I like to do, this would be like not someone that's even worth putting through pre-processing. But yeah, I have had instances where the MRIQC didn't look great. Maybe it was borderline, but then the fMRI prep looked good. But that's more borderline cases versus participants that are like very obviously exclude.
Yeah, that makes sense. And then are you working with like adult data when you're doing this, or developmental data?
Yeah, when I've done this in the past, I've worked with adult data in samples that putatively have lower self-regulation, so smokers. And so that data had a little bit higher movement than your typical adult data set, but still within the range of what leads to a decent looking scan. I have looked at some, not with MRIQC, but with different software. I've looked at some diffusion data from the ABCD data set. And yeah, your decisions might, your threshold for what's a usable scan might change a little bit, so you're not throwing out so much data. But that's kind of each individual lab decision to make of how stringent do you want to be with these thresholds.
Yeah, thank you.
Yeah, of course.
I'm curious if you have documentation of the artifacts that you listed in the quality rating. How do they look like?
Yeah, so we can skip. Yeah, let's skip forward a little bit and I can show you. So yeah, so one of our reviewers basically said the same thing, like show us some examples of bad data. And so I put together this supplement to go, well, now I'm going back and forth with the journal of where each figure goes. So some of these figures might end up in the main. Anyway, it will all be in the paper. And so I have some data here. These are, if you look in the curriculum, if you look on the Jupyter Hub, in the curriculum folder, in my folder, in the README, there are links to these, I think. If not, let me know and I'll drop them in the Slack. Because some of these are a little bit subtle. And so, let me see if I can, okay. So this is an example of a subject that I would exclude for motion ringing. So you can see, if you look closely, if you have kind of resolution, not on the projector, actually. If you pull it up on your laptop, there are faint rings across basically the entire brain caused by excessive motion during the acquisition of this T1. And it's to the point where I would consider this a potential exclude. And then if we look over here at the supplement, we can see what this looks like. And yeah, so here we can see how that might look in the background image, where you can see like these, you can't see it very well on this projector, but there's basically faint rings coming off of here from the brain, again, from motion. And so again, that would be this one is not quite as severe as the other one that I just showed you, but that would be something that I would watch out for. Also, so in the extended data, I have like 3 examples of participants that I might exclude. This is another one that I might exclude from a functional scan, where this is where the sinuses meet the brain. The brain gets a little bit distorted because of that interface between the air and the brain. And so it can cause the brain to kind of like be pulled out of the skull like that. So you would expect the brain to round out like this, but instead the signal gets pulled out. This ranges from mild to severe. This is a pretty moderate case of it. This is something that's it's susceptibility distortion, and it can be fixed by fMRI prep, especially if you have a field map. But if it's really, really severe, it might not be fixable. So this might be one where you take a note of it to check the fMRI prep output to make sure that it got adequately addressed. And something that I haven't talked about yet is ghosting. So ghosting is when you have an outline of the brain that shifts, and it is caused primarily by, again, MR physics, and it's rarely visible in this bold image. But it is visible in this one because it's such a severe case, where you can see there is basically a wrinkle in the brain right here. And that's because the outside of the outline of the brain basically shifted, and it's present in enough of the time points that it shows up on this average. So this would be a potential exclude. And so these are basically 3 scans that are pretty bad. And then the supplement is just examples of other more benign artifacts. So this one's kind of fun. It's a little bit hard to see because of the defacing that happened here, but you can see the MR glasses there. That's benign. It gets removed and the skull gets stripped, but you can still see it. There's also a fair amount of noise around here. Again, pretty benign. I wouldn't really worry about it, but this is a good example of noise and of these MR glasses. Sometimes you also might see the head pads that you use to stabilize the participant's head. You might see those. What else have I seen? I think that's it. But yeah, those are some common ones that you might see. I showed you this. These are rings caused by motion here. This is an instance of wraparound. So you can see it's a little bit hard. I want to find an example of this that doesn't have the defacing. but essentially you can see how part of the face basically is cut off by this frame of view, and it wraps around to the back of the brain. So this is usually okay, as long as this wraparound doesn't go too far into the brain. And this is caused by the scan operator usually not assigning the field of view properly, or getting it a little bit too close to the brain. This is a eye movement artifact where, because the eyes are always moving, you can get some eye spillover here where the movement from the eyes kind of moves out. This is fine right here because it's not in the brain, but it also comes back into the brain as well. So in this instance, it's not too severe. I would look at the mosaic view of the brain and look to see if that area of the brain looks noisy at all. You're not going to be able to see this, but there is more ringing in the brain where the little where this is. So there's faint rings there. And these are all annotated at the bottom here with a potential diagnosis of what's going on and which slices to look for. We talked about intensity non-uniformity. This is just where part of the brain is a lot brighter than the rest of the brain. It's fixable, but that's a really nice example of it because the rest of the brain also looks really, really nice there. Yeah. This is an example of what excessive noise looks like in the mosaic view rather than the background view. So this is a scan. So this scan and this scan both suffer from the same problem, which is noise in the background. It just presents differently in the different visualizations. And so if I saw this scan up here, I would go and look at this visualization to confirm that it's not too severe in this visualization before I made a decision about whether or not to include or exclude that participant. Here's another example of the ghosting that in the fMRI image that I just showed you. It's a little bit clearer here where you can see clearly this outline of the brain here. That's not supposed to be there. And then this is a potential way that you can diagnose excessive motion is by looking for symmetric highlights on the edges of the brain. So you can see that both the front and the back have basically a lot of variability. And so that can be indicative of motion. And that can, oh, and here's more ghosts this time, just in the background view. So you can see here that there is part of the, you can see an outline of the brain out here where it's not supposed to be. Oh, and then this one, the brain is upside down. That one's an easy one. This usually doesn't happen. What data set did this come from? This came from one of the open neuro data sets. And actually we're lucky that MRIQC even ran on this. Sometimes when a scan is upside down, or if you have like a really restricted frame of view issue, MRIQC just won't run. And then we have more intensity non-uniformity. So it's a lot brighter on the top of the brain here. We have some examples of what motion looks like in our carpet plots, and then what respiration looks like in our carpet plots. And I don't know, I think this might be coil failure. So we can see that there's like this large change in our carpet plot from like right here on, but there's really no associated like spike in motion. So this is just suspicious, and I would go look at the raw image to see if you go and look at the raw image, you can maybe see something that happened during the scan here. And we haven't done the diffusions yet, so I'll skip those. Any questions?
Question on mine.
Yeah.
Is there a way to export specific metrics of interest like FD from MRIQC to the second file?
To export them?
Yeah.
Yes. So along with these, along with the visualizations, we also get, so we have our MRIQC folder here. This is just on the Finder app of my Mac. So we're in our MRIQC folder, which is where I saved all of my MRIQC HTMLs as it was running. And it's just within these subject folders. Again, this is bids-ish. So you can navigate it the same way that you would a bids directory. So if I want to look at the functionals, I'd click func. And we have TSVs of all of the different image quality metrics that you could possibly want. And then you can, this is easily ingestible by pandas, Python, R if you want. Is that good online? Okay. I'll take that as a yes.
That MRIQC can be used as like a quality metric for like every stage of the data handling. So I'm thinking like if you do it for MRIQC, then you run MRIQC and you want to run it again and see like the data quality improved after, or how the data quality improved after MRIQC. What images do you plan to for the input?
So I may have misspoken, or maybe we're misunderstanding. You should do QC at every different step. MRIQC is not necessarily the tool that you should use for that. fMRI Prep makes the visualizations for you. So a lot of these visualizations, fMRI Prep will make also just on the pre-prop. John?
For the dimension like FD versus a 6-parameter motion plot, any thoughts on that, or I don't know if you have any insight into what the MRIQC folks are thinking about that?
No, but if you post on NeuroStars or on their GitHub, you might get some debate. Yeah. So I'm going to go back to where we left off on our ratings here and just go through the rest of this. Yeah, so rating widget. There we go. So there's some best practices for rating the reports. So we usually say use the non-deface scans when ethically possible. So obviously you can't share the scans when they have faces on them. Sometimes you can't upload them to your HPC if they have faces on them, depending on the security there. And if they come from OpenNeuro, they obviously don't have faces. But I'm just going to show a quick example of the differences, how these, basically what you get by working with the non-defaced images. So here is a scan that is defaced. So the face has been removed. I think this one is probably with, yeah, so I don't know what algorithm does this. PyDeface, one common one will just slap a rectangle over the face. I don't know what does this jagged thing, but look at like how much information you're losing right here. Like you can't see if there's any eye spillover. You can't see really well if there's any sort of wraparound, versus if you go here to this MRIQC scan that was the scan that was processed through MRIQC and then posted online. So this is fine, even though it's non-defaced, it's just the image, so it's fine to post. These ones are fine, at least check with your IRB. You can see that there's eye spillover here that we're missing that we would be missing if we had defaced the scan. There's preliminary, there is a recent paper out by Celine and Oscar, my co-authors, showing that it doesn't have a huge impact in calculating the IQMs, so calculating the image quality metrics, defacing the image and removing that information. But there is a small effect of the scores that you assign to these scans. So when you're doing the manual rating of defaced versus non-defaced. So when possible, and this is easiest if, say you have flywheel, the flywheel gear can run before you run defacing gear, and then you get these. use a big screen when possible, try not to use a projector, turn up your backlight so that you're getting, as bright as possible, have more than one rater for as many scans as possible, because then you can calculate the inter-rater reliability. Especially if I have a new RA who I'm training, I'll overlap with them more than if I was, say, trading off with Kelly trying to QC a large data set. And if you find something suspicious, you can look at the raw data. This is a little bit more time consuming, but you can find, like, there are some things that you can't tell from a visualization that you can tell by flicking through the time points of the raw data. Some lessons learned, don't do them all at once. If you are doing a data collection acquisition where you're scanning all the time, it can be really tempting because you're scanning all the time to not, put it off and then do them all in a couple months. That's somewhat unpleasant. Get cozy, put on some music or an audiobook. Your back will hurt if you aren't sitting ergonomically. Take eye health breaks every 20 minutes. You should look 20 feet away for 20 seconds. And really use this rating widget. I scored a lot of reports prior to the rating widget being implemented, and the rating widget is just very nice. So you're always making sure that you are keeping proper track of your scans. And my dog likes to join me and rate them with me. So, and then when you're all done doing that whole process of rating all of your images, you can compare your ratings with your co-rater. You have your JSONs, you can pull those into Python, and you can look at if you and your co-rater deviated too much. What you should be looking for is whether or not you agree on whether to include or exclude the participant. If I rate something a 3 and Kelly rates it a 4, I wouldn't quibble about that. That works. If I rate something a 4 and she rates it a 1, then we have to discuss that and figure out, like, why does she think that this is an useful scan and I think it's excellent. Add your ratings to your participants.tsv. You can add an extra column labeled MRIQC ratings, and then anyone who's looking at your data set will know those are your MRIQC ratings. It's really helpful if people share bad images. A lot of people will only put their curated data sets on OpenNeuro, which is great, but that means that people who are doing quality control work, who are trying to improve the quality control tools that we have, they don't have examples of bad images to work on. So I can't see if, like, fMRI prep can fix, like, really, really bad susceptibility distortion if I don't have any examples of really, really bad susceptibility distortion. And I then can't also show people examples of what a bad scan looks like with specific artifacts if I just can't find those specific artifacts in these data sets. And then optionally, if you collect enough ratings, if you have a lot of scans, you can try to train some sort of QC classifier. There has been success doing this. However, it usually only works within site. So I would only be able to predict the quality of other scans from the CHN using my scan parameters, which is not, other labs might not be using my specific scan parameters. So this is something that the MRQC folks have worked on a little bit, but it's kind of a difficult problem. And if you figure out something suspicious, please tell your scan facility so you can save other people from collecting bad data. And okay, and then I already showed you a lot, and I thought this was going to go a lot faster. So I was going to have people look through some of the PNC data set and see if they found anything interesting, which we can still do, but there's only like 10 minutes left. So yeah, okay.
So on Zoom, there's a question, is there a command to extract our widget quality control ratings to a CSV file, or should we write some code to extract the HTML files?
So the widgets, your quality control rating. So I say this scan is a 1. That gets saved to actually a JSON file, just this fancy little text file. Right here. So this is an example of the JSON folder. JSON is very easy to work with in Python. Where'd it go? So you can write, like, I don't know, 5-10 lines of code that will extract my rating from this JSON, and then I can, it looks at how much time you spent. So you can, figure out if your RAs are spending a lot of time on it, or if they're very, very efficient at it, alternatively. So yeah, really easy to pull the information from this and make a nice CSV of it.
Another one is it possible to get the scanning SOP resources?
Yes, those are all included in this. There's a link to this Google Doc in the curriculum folder, Hagen, MRIQC, README. There's a link to this. It's in this. So it's in this document. It's also in the Nature protocol paper. There's also a link to that. And there's more information about standard operating procedures in the MRIQC paper as well, if that's something you want to dive into.
So is the frame-wise displacement the primary values that we use to decide the quality of data? How do we know which metrics that we use to decide the quality?
Yeah, frame-wise displacement is an easy one because it's very clear how to interpret it. If you're interested in the IQMs, I would look at Esteban 2017 in, there's a good paper on some more of them, I think. Esteban 2017 in, so this is the paper, no, that's not it. I think, yeah, that one, +1. Yeah, you can look at that for more information on the IQMs, and which ones tend to correlate with manual ratings. JB?
I guess one overall remark is that if you can check that your results remain robust with different QC level of things, I think that's a very good thing to have. So
yes,
just like a little bit on the side of analytical flexibility, what is robust with the QC thing, right?
yes,
if you get the result when you have a very hard QC, but you don't, if you just include more those 5 subjects are a bit like that, then you have to be, be careful, I guess. I mean,
yeah, that's what I wrote my GRFP application on, was doing exactly that.
Okay.
So yeah, if anyone wants to do that this week, let me know, I can help.
So yeah, that's one thing. The other thing that I just saw, I mean, at HBN, and there's a paper on that, and I put it in the things that there's a very nice method to actually use those ICM measures to correct for site effects.
Oh, okay.
So instead of having the sites as a natural, measure of the site, so well, many studies are actually cross sites, many are not, but correcting for the side effect is always like a hurdle.
Yeah.
There's a really cool paper by I'll put it in the slide.
Okay.
On how to actually correct for not only I think actually not only side effect, but they basically have a way of and that may be a bit computationally expensive, but of looking at those ICM as prediction of the sites and correct for that as there's several variables, not only the sites from that. So it's pretty cool.
Yeah.
It looks like it's working pretty well as well. I don't know how computationally expensive it is, so it's MCMC, so it might be.
Cool.
Are there any extra things that we should look at for diffusion, or is it exactly the same?
Yeah, diffusion is very similar. Here's a diffusion. So
Something more or less.
You have a very similar, this is very similar to your bold average map, except it's the fractional anisotropy map. And what you're looking for here is looking for, this is all in my paper, so you can also read through my paper for like a more in-depth explanation of this. Ariel, also a good resource for that. You're looking for like being able to see the white matter anatomy really clearly without excessive. So you see these little, there's little speckles right here. In some scans, there will be little speckles all over the entire brain. That's not great. On the edge here is fine. So yeah, you're looking for like clear white matter anatomy and some contrast between the white matter and the gray matter. Here is your MD map. You're looking for much the same. You're looking for like nice clear ventricles and darker darker white matter here. And then this visualization doesn't, you can skip this visualization because this is for the B0 shell. But this is the background of the diffusion image. So this is very similar to the background of the fMRI and of the structural MRI. So you're looking for noise here. And then here, what you're looking for, this is the standard deviation just plotted onto the diffusion image. So you're looking for being able to see this white matter anatomy nicely. And then it has all of the, it has one of these for every shell that you have. So I think I have a bad example somewhere here. So this is an example that I would not be too happy with. So you can see how there's a lot, these are not like super clear white matter tracks here. They're a little bit fuzzy on the edges. Again, here, there's not a lot of contrast and parts of this MD map are a little bit light. Nothing super wrong with this background image. It's a little bit noisier in the background. You can tell it's not bright yellow, it's this teal color, which by itself wouldn't be too concerning, but then paired with this doesn't look awesome. You're not seeing all of the white matter anatomy, and you can see that there's some dropout here where there's no signal. And okay, this is not the scan I was thinking of. Still not great. But there's one that's just like really bad. Yeah, here. You can see that you can't see any of the white matter anatomy. There's no tracks there. There's only variation along the edges. So this would be a pretty bad scan. I have a collection of, if you come talk to me later, I have a collection of diffusion scans from the Healthy Brain Network. Because it's kids, there's a couple of pretty bad scans in there, so I can show you some more from there. Yeah, of course. John?
Just like a comment, reiterate something you said, I think. Like McKenzie is not waiting, grouping everything together to do it together, I'd say early and often is really what you want to do. You don't want to wait and the experiment collected.
Yeah.
50 subjects, and then you go through your K of work. You really want to do it perfectly, starting a pilot, catching any systematic problems with the scanner at that point, and then looking early so you fix anything early that is so bad that it needs to be fixed, right? Yeah,
you're gonna save your RAs a lot of work from having to go through. It's kind of sad going through a bunch of bad data because you're like, oh, okay, we're gonna have to work really hard to salvage this when a lot of people put a lot of effort into collecting it. So Yeah, and I hope MRIQC makes it a little bit easier to do it frequently. And if you have,
That's more like, I also sort of heard from a lot of people expressing, it's like, how do I know if something's just poor image quality? Or if it's just like, the brain is the brain, everyone's brain looks different. Or usually one of the rules that I was taught when I first started doing QC is that if you see something that looks geometric in terms of patterns, stripes, random lines, speckles, things like that, patterns that are not natural, like rings, circles, things like that, they're artifacts probably related to your data acquisition itself. Obviously there's other things that, a lot of these background images will catch, or it just looks like the raw image itself. But a lot of these like geometrically related things, they're probably related to the actual image quality or acquisition quality. So those are pretty good things to keep in mind.
and as you do more, you get an eye of what's normal and what's not. That's kind of frustrating to hear, but you just kind of like get used to what weird looking brains look like. And you get a good eye for picking out the weirdos. And so,
Well, like Kelly said, I think that anything that like, when you have looking at these plots, think of like person versus machine, and we have to mention that Nicholas's log, that's a really good place to look at. If the noise or the quality problem is coming from, this is it's coming from scanner. That's really different.
okay, it's 5. So unless anyone has anything else. Thanks online, folks. See you around.
Thank you.
Thank you.